I didn't read very much. Sorry.
Wrote notes this time. I'm out of practice. It's been 4 years. Today we're at Lauren Fuess's defense. Lauren received her Bachelor of Science degree in 2012 from the College of Charleston. She then spent a year in Jamaica on a Fulbright Award and started her PhD at UTA in 2013. One of the first things I remember about Lauren when she interviewed back in 2012 was how eager and excited she was about RNA-seq. So she joined my lab with that intent to do a lot of RNA-seq, and we had just begun doing transcriptomic work, so it was a good fit. But I was quickly discovering that I really wasn't a huge fan of some of the data outputs, and if I saw one more Venn diagram of DEGs and PCAs of log fold changes, I was gonna just can the whole thing. So I tasked Lauren to show me something new and show me something exciting, and especially to show me something that targeted our interest in pathways of immunity. And so, boy, did she ever. She really excelled at finding the best analysis, and you'll see in her talk, the best analysis for every data set and for every experiment to suit that experiment and to test our hypotheses. She quickly became our bioinformatic guru in the lab, and with help of her committee members, of course, helped a lot of our incoming students. So she was able to come up with these great analyses, as well as to relate the protein work and our organismal phenotypes to transcriptomics, where I felt was really where the heart was. Over the past few years, and with the help of the NSF GRFP, she was able to analyze multiple data sets, run multiple experiments, and publish 4 first author papers and 2 second author papers. This amazing record has led to 2 postdoc offers, one in coral reef restoration and one in the thermal regulation of insects. So you can bother her later and see where she is on this item. Over the past 5 years, Lauren's really matured into an amazing scientist, and I'm excited to see where her future goes. I'm also really excited for this transition from being my student to my colleague. Okay, Lauren.
Thank you, Laura, for that introduction. Thank you all for being here. I'm really excited to talk to you today about the work I've done over the past 5 years, looking at ecological immunity on coral reef systems. So being biologists, I'm sure you're all very well aware of the importance of coral reef ecosystems. Not only are they incredibly biodiverse, but they also provide numerous important ecosystem services, such as serving as a source of coastal protection, as well as generating millions of dollars a year for coastal communities in ecotourism and fishing industry revenue. Finally, they're also a source of untold natural products, which may have medicinal or industrial applications. However, if you watch the news, I'm sure you're also probably aware that coral reefs around the globe are in dramatic decline, and the media tends to focus on these declines through the lens of thermally induced bleaching or breakdown of the symbiosis on which corals depend. However, what doesn't get a lot of attention is the role in which disease has to play in these declines, and it's arguably as large or larger than the role of thermal stress. Coral diseases are rapidly increasing across the globe. They first burst onto the scene maybe 30 to 40 years ago, and now we have almost 30 different types of disease which have been described in the literature. Additionally, they're very poorly understood. For those of you familiar with immunology, very few of these diseases we have fulfilled Cox postulates, meaning we know what causes the disease. And what complicates this is things like corals have very limited ways of showing us that they're sick. I'm showing just some of the different diseases here. This is dark spot disease. This is white plague and white band. As you can tell, these are they're both turning white. So that makes it difficult to distinguish diseases based on anything but these vague symptoms. The other interesting thing about coral diseases is that almost every single species of hard or soft coral is infected in some way by a disease. Additionally, several of these diseases, such as white plague, which I'm showing down here, affect a very broad range of species. So you have up to 20 species affected by the same disease. So it creates a really interesting system, and it really exposed, when it first became a problem, the gaps in our knowledge of the coral immune system, which up until about maybe 20 years ago was largely a black box. However, in the past 20 years, we've been able to really piece together what the coral innate immune response looks like. And it turns out it's really highly homologous with even the innate immunity which humans like you and I have. Corals have conserved pathways like toll-like receptor signaling, which results in antibacterial activity, complement cascades, which can lead to phagocytosis of pathogens, as well as invertebrate unique pathways like the melanin synthesis pathway, which deposits a tissue which can be used to encapsulate pathogens for later phagocytosis. So while we've really increased our mechanistic understanding of the coral immune response, what's lacking is our understanding of how these types of responses interact with the ecology of coral reef ecosystems. So for my dissertation, I decided to kind of land in that area and ask, how does this translate to ecology in a changing environment? So the field of coral ecoimmunology is a diverse field, but pretty tight-knit field. Ecoimmunology, when I say that word, basically what I'm referring to for the context of this talk is studies of how immunity affects the ecology of systems. So how and why immunity varies and how that affects organisms as a whole. And we can look at this from a number of different lenses, including looking at inter and intraspecific variation in immune response and how that affects communities, as well as life history trade-offs. So down here I'm showing a figure from a former postdoc of our labs, Jorge Pinzon, where he looked at a variety of different life history traits and how they affect the immunity of different species. So here we have a massive coral and a branching coral, and these two corals vary in a lot of different ways in terms of life history, and their immunity also varies. We can also, from an eco-immunological standpoint, look at the effects of climate change on coral immunity and coral disease. So this figure here is showing the classic disease triangle and looking at the effects of temperature in particular on the host's immune response, so increased susceptibility as well as changes in behavior, and then on the pathogen. So increasing temperatures can also increase the virulence of a pathogen potentially. So my dissertation predominantly laid within these realms and sought to answer quite a few different questions of ecoimmunology on reefs. So what I'm going to start with is just a brief overview of the first couple chapters of my dissertation, just to give you an idea of how we did things, and then I will focus in on the last two chapters today. So again, I'm showing that classic ecoimmunological disease triangle. The host, the environment, and the pathogen, and where they intersect is disease. My research has predominantly focused on things from the host side and used two different types of analyses to look at this. The first being our traditional biochemical assays, which give us an idea of protein activity of a variety of immune responses. This includes antibacterial activity, antioxidant responses, which are not only a part of the stress response, but also a part of the immune response in corals, as well as that melanin synthesis cascade and propionol oxidase cascade that I mentioned before. I coupled this with transcriptomic analyses, both in terms of simple differential expression analyses, as well as more complex pathway-based analyses and co-expression-based analyses. And so these methods together and separately have allowed us to gain a lot of insight into the coral immune response. So for my first research chapter of my dissertation, I focused on using these types of methods together to look for new candidate immune genes within our model coral Orbicella fabulata. So what we did of RNA sequencing and bioinformatic assays, we correlated them to one another to identify new novel immune genes. And this down here is just showing an overview of that, of our different immune genes signaling, receptors, signaling, and effectors, and their correlations to different types of immunity. So this chapter allowed us to identify new targets for looking at when we study the coral immune response. I then took this data one step further in my next chapter and looked at pathway-based trends in gene expression in 4 species of Caribbean coral, which range the gamut from extremely disease susceptible to disease resistant. So using pathway analyses, we were able to characterize the response of these species to an immune stimulation and look at what pathways they were increasing or decreasing based on that immune stimulation and compare them across each species. And what we found was that our susceptible species, such as Orbicella fabulata, increased expression of genes associated with apoptosis or cell death in response to that immune stimulation, which likely results in cell and organismal death. In contrast, our resistant species, such as Parietes, Astroides and Parietes parietes, upregulate autophagic pathways, such as which allow for plastic responses. So autophagy can be involved in the immune response of corals, as well as just general cell turnover, allowing them to initiate this plastic response to a challenge and hopefully resist and adapt. So this created a new paradigm or shift in our thinking of how corals respond to disease and what contributes to disease resistance and susceptibility. We took that data and then moved forward with a collaboration with colleagues from the University of Boston, and we used that data to elaborate an NF-kappa B signaling pathway within cnidarians for the first time. So using that pathway data, as well as a lot of different techniques that the Gilmore Lab at Boston University helped with, we were able to first describe toll-like receptor to NF-kappa B signaling in a cnidarian. Shown here is a full description of that pathway using my data, with genes which are expressed in yellow and genes which are not expressed in blue. So this was the first description of that pathway. So for the majority of my talk today, what I'm actually going to spend the most time talking about are the last two chapters of my dissertation, which look at trade-offs in the context of immunity. And so what I'm going to define trade-off as, for the purpose of this talk, is a context in which one trait, such as immunity, increases at the expense of another. I'm going to talk about two different types of trade-offs. Immunity and life history trade-offs. So this diagram here kind of depicts what I'm talking about in that terms. So an organism has a set limited amount of resources which they intake within a day or their lifetime, and they can then allocate those resources to either reproduction, immune function, or growth and cellular maintenance. So oftentimes you'll see one of these, such as immune function, increase at the expense of another. So my first chapter will deal with this in the context of natural disease. Then I'll switch gears and I'll talk about trade-offs between immunity and symbiotic relationships. And by this, I mean symbiotic relationships, particularly corals, by nature have overlap with the immune system. A symbiont, particularly endosymbionts, is non-self, but somehow living within the cell, and therefore must suppress or evade immune responses in order to establish and maintain that relationship. And so for my last bit of my talk, I'll talk about this trade-off, and this is just kind of a preview. What we're showing here is anemones, which are without symbionts and with symbionts. And as you can see, there's a trade-off between immunity in terms of that NF-kappa B signaling, where anemones with symbionts have much less NF-kappa B signaling. So just like I said, part one, I'm going to talk about immune growth trade-offs in the context of cnidarian disease. And then I'll switch gears and talk about relationships between immunity and symbiosis through the transforming growth factor beta pathway. So starting with part one, this chapter begins as part of kind of out of a casual observation of a colleague of ours in the Florida Keys. She noticed corals of the species Unicea, or genus Unicea, were getting infected with a strange disease which caused them to turn black. So now the disease is called Unicea black disease. That's how we name coral diseases. And so this piqued our interest because these black lesions shown here on the infected individual, and this is a healthy, are typically indicative of melanization or deposition of that melanized tissue, allowing for phagocytosis of particles. And so we were really interested in this as we've been long trying to describe this pathway on both the protein and transcriptomic level. And so this seemed like a great data set to look at. And in fact, our researchers were able, colleagues were able to confirm that that phenotype is the result of melanin deposition on the surface of these Unicea corals. So we went into this trying to use transcriptomics to understand the immune response of a specific species of Unicea, Unicea colliculata, to this disease with a particular lens on the melanin synthesis pathway. We also wanted to, since we're doing full transcriptome, characterize the physiological consequences of this response to disease. So what we did was a former PhD student from this lab, Whitney Mann, traveled to the Florida Keys and collected a bunch of different samples of Unicea But in particular for this project, she collected 3 healthy and 3 disease samples of Unicia colliculata, and she flash froze them on site, brought them back to the University of Texas at Arlington, and extracted their RNA, and then sent them to UT Austin for library preparation and sequencing. Once we had the reads back, we then went through a standard read processing and assembly pipeline using quality and adapter trimming in Trim-O-Matic, and then we assembled the transcriptome de novo using Trinity. This species has no existing resources, so we had to do everything from scratch. And then what I'll talk about today are coral-only transcripts. So what we did was we took that de novo transcriptome and we compared it to what we have a meta coral genomic resource. It's made up of a bunch of transcriptomes and genomes from a number of different coral species. And we compared our de novo assembly to that to obtain just the coral-only reads. We then analyzed that coral-only transcriptome using differential expression analysis in the CuPlink suite of software. We annotated the de novo transcriptome using BLASTX, and then we used some complex differential expression analyses and gene correlation analysis using the R package weighted gene correlation network analysis. And what this does, if you're not familiar with it, is it takes all of your gene expression data and it basically builds groups of genes which are doing the same thing or ideally co-express. So if they have similar patterns of expression across all of your samples, they're put together into what's called a module. And then you can then take those modules and correlate them to traits of interest. In this case, I'll talk specifically about their correlation to whether or not a coral is diseased. You can then further analyze those groups of genes using gene ontology analyses. My favorite technique for gene ontology analysis is GO-MWU, which is a rank-based assessment of gene ontology, and tends to get you a lot more detail than things like Panther or David. So just starting with the differential expression analysis and gene ontology analysis of that, what we found were multiple immune-related differentially expressed genes, mostly associated with. So we found lysozymes, peroxidases, macrophage receptors, all of these things that are associated with that phagocytosis, which is exactly what we would expect. The corals are heavily melanized, so what we're seeing likely is a genetic signal of a phagocytosis of melanized particles, so that late stage immunity. Gene ontology enrichment was actually in congruence with this. We found one significant gene ontology term, biological adhesion, at least for the biological processes. And most contexts associated with this were upregulated or increasing in expression. And we believe this is linked to that melanization and phagocytosis, as adhesion is extremely important in phagocytosis and melanization. So this was pretty standard, exactly what we were looking for. Where things got really interesting was when we began to look at our gene correlation network analyses. Our network was comprised of 29 modules, ranging in size from just under 200 to just over 3,000 genes. And when we correlated this, we correlated it to a bunch of different things, but what I'm going to focus on is this column here, correlation to disease status. We found only 2 modules which were significantly correlated to disease status, both of which were over 3,000 genes in size. This were module 19, which I'll talk about first, which was positively correlated to disease, and module 4, which was negatively correlated to disease. So we wanted to look at these 2 modules specifically and use that gene ontology enrichment to see what processes were in there. So pay attention to these figures because I'll show you a lot of them throughout the rest of the talk. This is the output of that GO analysis I was talking about, GO-MWU. What you have here is a hierarchical clustering of significantly enriched gene ontology terms. Branch lengths over here indicate how dependent each of these terms are on each other for their significance. So when you have terms like these 3 here, which are connected by a very short branch, that means that their significance is caused by a small or whatever size group of genes dictate their significance. So they're highly dependent on one another. The p-values are shown up here, and they'll adjust slightly to make the figure legible, but I'll make sure to point out any terms which aren't shown that are less than 0.05 and are relevant to my discussion. So starting with the first thing we notice about this module is that it was enriched for a lot of terms associated with immunity. Regulation of wound healing, regulation of inflammatory response, as well as defense response, endocytosis, and several other genes. So remember, this module 19 is positively correlated to disease. So these terms are increasing with disease, which is, again, exactly what you'd expect. Immunity, and particularly these types of immunity associated with late-stage immune response, are increasing. This was coupled with quite a few terms associated with ERK and MAP signaling, G protein coupled signaling, things associated with immune signaling. So we characterize this module 19 as our immune and signaling module, and it is increasing with disease. So immunity and signaling are increasing with disease. However, when we looked at module 4, which was decreasing with disease, we saw a completely different story. In fact, we saw that this module was enriched for things like cell division, cell cycle, DNA replication, all of these processes associated with cell growth and division. And remember, it's negatively correlated to disease. So module 4, which is negatively correlated to disease, became our growth or cell growth and division module. So this led us to come to an interesting conclusion. We had module 19, which is positively correlated to disease, enriched for signaling and immune processes, in contrast to module 4, which is negatively correlated to disease and enriched for cell cycle processes. This created an interesting disease model where you see immune response increasing at the direct cost of cell division. And while this wasn't completely surprising, we've had colleagues document the cost of disease in natural corals in terms of reduced fecundity and growth. This is the first transcriptomic evidence of this trade-off, which has long been theorized by various different ecoimmunologists in other systems. So to summarize, we documented a strong immune response of Unicia paniculata to this disease, which was characterized by that late-stage immune response of phagocytosis and melanization. However, this response came at a cost, and that cost was cell growth and division. And this provides a really interesting ecological kind of conundrum. Particularly soft corals like Unicia are size-dependent reproducers and asexual reproducers. And what that means is the larger they are, the more output they have each reproductive season. So when you have something like disease, which not only kills quite a few individuals and reduces their size through tissue loss, you're also going to see a decrease in growth, which is going to reduce the ability of these individuals and communities as a whole to recover following a disease outbreak. Further nail in the coffin is that these species are extremely dependent on self-seeding, so reproduction of at the location, to recover following a disturbance. So this kind of chapter, we walked away with the conclusion that when we consider disease, we need to think beyond just the direct mortality of individuals and consider things like immune trade-offs when we're predicting reef futures. So that's the end of that story. Next, I will talk about how symbiosis and immunity interact through the lens of the transforming growth factor beta pathway. So symbiotic relationships exist on ecosystems throughout the globe, and they range the gambit from mutualistic, where both partners are believed to benefit, to parasitic, where one partner benefits at the expense of another. On coral reefs, they're the foundation of cnidarian success. Particularly in hard corals, these relationships are what allow the organisms to thrive. Hard corals are dependent on relationships with single-celled dinoflagellates known as symbiodinium to meet their nutritional needs. And this relationship varies considerably both within and between species in both the identity of these symbionts hosted and the density at which they're hosted. And what's really cool is that variation in this relationship has been shown to have significant implications on the tolerance of these corals to bleaching or thermally induced stress and breakdown of that symbiosis. However, when I began my dissertation work, I was really surprised that no one had looked at this in the context of immunity. As I mentioned at the beginning of my talk, symbiosis by nature has overlap with the immune system. Symbiodinium, being endocellular symbionts, must somehow either be recognized by or suppress and evade immunity in order to establish and maintain that relationship. What's interesting is new evidence has shown that perhaps, even though they're mutualist or believed to be mutualistic partners, they may share mechanisms in doing this with parasites. In particular, new evidence has suggested that perhaps they manipulate the host's own transforming growth factor beta pathway. So TGF-beta is a signaling cytokine, which is found throughout metazoans and has a lot of different functions, especially in higher vertebrates. But in cnidarians, it's believed to be immune regulatory predominantly. And what it does is it prevents an excessive immune response. So it suppresses things like the melanization and antibacterial response. Interestingly, in a variety of different systems, it's manipulated by parasites to promote host tolerance. So basically, a parasite will come in, hijack this pathway, and increase its activity in order to cause the host to suppress its immune response and allow the parasite to continue its infection. New evidence in coral systems and cnidarian systems has found homologs to this pathway in both our model anemone, Exaptasia, as well as my model hard coral, Orbicella fabulata, shown down here. Additionally, that work in that model anemone has shown that this pathway directly contributes to the susceptibility of corals to bleaching. So when you take anemones and place them in exogenous TGF-beta, they are less likely to undergo breakdown of symbiosis or bleaching following heat stress. Also, that paper found that perhaps somehow symbionts were increasing activity of TGF-beta within the anemones. So for the last section of my dissertation, I wanted to look at this pathway specifically and look at how it might affect symbiotic relationships in corals and the ecology of these systems. So what I did was I took a multi-experiment approach to understand this relationship between symbiosis, transforming growth factor beta, and host immune response. I started with an experiment that just wanted to look at the effects of TGF-beta on host gene expression, and particularly immunity. And then to add ecological context, I coupled that with a manipulation of the symbiont density within a coral host to see how that affected not only host transforming growth factor beta activity, but also host gene expression and immune response. For the purposes of today's talk, I'm going to predominantly talk about experiment 1. As experiment 2 is being currently reanalyzed, I will give you a small preview of that data to provide ecological context to my findings of experiment 1. So experiment 1 was conducted at the University of the Virgin Islands in St. Thomas. What we did was our colleagues went out and collected 5 large colonies of Orbicella fabulata from a local reef, and they were split into 8 replicate fragments of roughly the same size. So this is about as close as you can get to genetic replicates within corals. Those 8 fragments were then divided between 8 different treatments, which resulted from a full factorial combination of our 2 different treatments. So we started with a pre-treatment to manipulate the transforming growth factor beta pathway within our samples. This was either exogenous transforming growth factor beta, which would enhance that signaling and kind of mimic increased symbiont density, and this was coupled with a seawater control. And then we had an anti-TGF-beta treatment, which would inhibit signaling of this pathway. And that was made in a rabbit antibody, so we used a separate rabbit antibody control to control for those effects. So we injected corals with these serums and then allowed them to incubate for about 2 hours, at the end of which we then pulled them back out and exposed them to a second treatment, an immune challenge treatment, using a cocktail of lipopolysaccharides and peptidoglycan, both of which are derivatives of bacteria, to elicit an immune response, or a seawater control. The samples were then preserved in RNA later, and portions were preserved in RNA later, and the entire fragments were brought back to UTA, where they were processed for proteins and for RNA extraction. Extracted RNA was sent to the Novogene Corporation for sequencing, where they were sequenced on an Illumina HiSeq with 150 base pair paired end reads. The reads were then processed pretty similar to what we did for the first part of this talk. We did quality and adapter trimming and trimmomatic. And then in this case, we have a reference Orbicella fabulata, which is the species we're using, coral-only transcriptome. So we align the reads to that transcriptome to get our differential expression read counts. We then did differential expression analyses in a different program. We did it in DE-seq2 this time, because that allows for much more complex modeling of differential expression and was much more better suited to our 8 different treatment groups than cufflinks. We re-annotated the transcriptome in UniProt just to make sure we had the most up-to-date annotations for all of our transcripts. And again, we did gene ontology enrichment using that Go MW. So I'm just going to talk about a subset of the contrast today. First, I'm going to talk about what we saw in the corals with just the addition of transforming growth factor beta or anti-TGF-beta with no immune stimulation. What we found is that there was almost no effect of manipulating this pathway when you have no immune stimulation. When you add transforming growth factor beta, so you enhance the pathway without immune stimulation, there are only 11 genes which were differentially expressed, which resulted in 3 significantly enriched biological process gene ontology terms. So we have those terms again over here. And this time they're colored because of the data. So red terms are increasing, blue terms are decreasing. And what you can see is this enrichment, positive enrichment of microtubule-based and cilium movement, which is often associated with TGF-beta signaling. So exactly what you'd expect. If you give them more TGF-beta, they're going to signal and process it more. Not really ecologically relevant. However, we also saw no effect of inhibiting this pathway. Only 6 differentially expressed genes and no enriched biological process terms. Where this became really interesting was when we then compared this to the effects of immune stimulation on top of manipulation of this pathway. So I'm just going to start by showing you the baseline immune response. So what this is it's showing you enrichment of biological process terms associated with the effect of immune challenge in those corals which were treated with seawater, no TGF-beta stimulation. And what we find is positive enrichment of 20 different terms associated with immunity. This includes NF-kappa B signaling, immune response, cytokines, defense response, all different kinds of immune response. And this is really in agreement with every other study we've looked at, whether it's in the context of immune stimulation or natural disease. So this is exactly what we expected to see and confirms that our immune stimulation worked. Where things get really interesting is when you compare this to the corals which were pre-treated with transforming growth factor beta. These corals, again, underwent treatment to enhance their transforming growth factor beta signaling, and we saw a complete reduction of their immune response. In fact, the only signal of immunity that's left is these 3 terms here that are associated with double-stranded RNA and type 1 responses. Additionally, we saw that they negatively enriched a bunch of terms associated with antioxidants, which if you remember from the very beginning of my talk, antioxidants are an important part of the coral immune response and the coral stress response. If you also remember from just a couple of minutes ago, I told you that corals which are treated with transforming growth factor beta have an increased tolerance of thermal stress. So what we think is going on here is that the corals are more tolerant of their algal symbionts and therefore reducing the expression of the antioxidants because they've kind of been deadened to these effects. Now, when we look at what happens when we add an inhibition of TGF-beta signaling with the anti-TGF-beta before an immune stimulation, we see that the immune response is actually preserved. So now we see 26 different enriched immune gene ontology terms, including things like toll-like receptor signaling, innate immunity, cytokines, interleukins, all your classic hallmarks of innate immune response. So what we see is when you knock this pathway out or inhibit it to some extent, you see a preserved or even strengthened immune response. So to summarize, we have little overlap in the gene ontology terms, which were significantly enriched between our different groups. So these are, each one of these is representative of a pre-treatment group, so a TGF-beta manipulation group, and then contrast between the controls within that group and the immune challenge group. And the greatest overlap you see here is between that seawater control and the anti-TGF-beta, or the inhibition control. So that further confirms that preservation of the immune response. In total, we actually saw over 1,500 contigs associated with those immune GO terms and apoptotic GO terms, which were significantly positively or negatively enriched. And so I wanted to look specifically at some of those terms and see what they were doing in comparison to the big process trends. And so what I'm showing here they'll show 2 different pathways. The first is inflammation, and then I'll show you toll-like receptor signaling. And I know this graph is kind of overwhelming, so let me walk you through it. Each one of these is a paired set of graphs per gene. And each one of these is an immune gene associated with inflammation. On the left, you have transforming growth factor beta effects. On the right, you have anti-transforming growth factor beta effects. The expression is shown on the y-axis, and then control and immune challenge on the x-axis. And then for each of these graphs, these red lines represent the effects of the treatment, meaning transforming growth factor beta or anti-transforming growth factor beta. So basically what I want to point out here is the fact that you see opposing trends in our treatment groups when they're exposed to immune challenge. So each of these genes decreases following immune challenge when pre-exposed to transforming growth factor beta, or in contrast, increases or stays the same in the anti-TGF-beta group. These genes, ADORA1 is associated with Fc gamma receptor phagocytosis and inflammation. ALOX5 is an arachidonic acids signaling molecule, and then P2RX7 is an extracellular ATP sensor, which senses accumulation of extracellular ATP at injuries and causes increased inflammation. When we looked at the toll-like receptor signaling pathway, we saw the same exact trends. So these are the same graphs I showed last time, just different genes. Same trends of decreasing following immune challenge when they've been exposed to transforming growth factor beta, and increasing following immune challenge when they've been exposed to anti-TGF beta. Again, these are DMBT1 is a gene which is regulated by toll-like receptor signaling. It's a scavenger receptor that I've previously shown in my very first research chapter to be positively correlated to antibacterial and oral. So it's one of our strongest candidate immune genes. And then you have toll-like receptor 2 and TRAC3, which is a intermediate signaling molecule in this pathway. So we see that these genes are doing exactly what we'd expect based on that gene ontology. So pathways and individual genes are all doing the same thing. To summarize, TGF beta enhancement results in almost no effect when you don't have immune stimulation. It increases ciliary and microtubule action, which we would expect under increased TGF beta signaling. In contrast, when you couple that with immune stimulation, we see a reduced immune response and reduced antioxidant response. On the other hand, inhibition of TGF beta with anti-TGF beta results in no effect without immune stimulation, and actually preserves or increases the immune response when you have immune stimulation. So this is great. We confirmed or provided significant evidence that TGF beta functions in an immune inhibitory way within Nigerians. However, we're still lacking the ecological relevance, which is why we did experiment 2. So experiment 2 was undertaken at the University of Miami in the Baker Lab, and they have this really neat method which they can use to increase symbiont density within coral cores using nutrient enrichment. So they took replicate cores of 4 different colonies of Orbicella fabulata, and for 1 month exposed half to nutrient enrichment and half to control conditions to manipulate that symbiont density within the cores. At the end of that month, we then exposed them to an immune challenge or control conditions and flash froze them in liquid nitrogen, did all of our transcriptomic work. These were actually sequenced at the UT Austin instead of Novogene, because this experiment was actually done before that latter experiment I just talked about. And we did full transcriptomic analysis. And I'm still analyzing a lot of components of this, but I will talk about some of the effects which we saw. The first thing I just want to establish is that nutrient treatment did increase symbiont density. So what I'm showing here is each of our 4 colonies of Orbicella fabulata. On the y-axis, you have symbiont abundance. On the x-axis, you have treatment. These purple bars represent control. The green represent our nutrient enriched. And what you can see is that within each colony, nutrient enrichment increases symbiont density. So this treatment served to increase the natural variation we saw in symbiont density. Because of this, we again used DEC2 to model symbiont density separate from our treatment effects, as depending on the colony of origin, a coral which was treated with nutrients did not necessarily have more symbionts than one that was under control conditions. And so what I'm going to focus in on is a subset of those genes which are significantly affected by symbiont density. Let me walk you through this graph because it is, again, a little bit daunting at first. Up here we have 4 genes which are involved in immunity, which were significantly affected by symbiont density. This is histone deacetylase 4, negative regulator of immunity, RIG1, which controls antiviral responses, SOD, superoxide dismutase, which is an antioxidant, and ZC3HAV1, also an antiviral. What I have below it is the patterns of expression for each of these 4 genes from that first experiment I talked about. So here is the response of these genes to transforming growth factor beta in experiment 1, and here is the response of those genes to anti-transforming growth factor beta. And what I want to get across to you is that in the case of histone deacetylase, which is a negative regulator of immunity, it's increasing with symbiont density, which is exactly what we would expect. It increases in expression when you add transforming growth factor beta, and decreases in expression when you add anti-transforming growth factor beta. In contrast, these 3 positive immune genes, RIG1, SOD, Z3HAV1, all decrease with symbiont density, decrease in expression with transforming growth factor beta, and either increase or are preserved with anti-TGF beta. So while this is correlative and not causative, it is indicative that perhaps there is some link between symbiont density and transforming growth factor beta, as these genes are responding similarly to both of these factors. So to conclude, manipulation of the transforming growth factor beta pathway has significant effects on corals, but only after immune challenge. And this is really actually quite interesting to me, and I'll get to why in just a second. Additionally, many of these transcripts associated with affected by transforming growth factor beta are similarly affected by symbiont density, indicating that there is likely a link between the two. And if there is a link between the two, this provides a really interesting new ecological trade-off. Disease has rapidly increased in the past 30 to 40 years, so it was not really a selective force probably before the recent century. So this idea that TGF beta affects corals under immune stimulation effects suggests that there is probably a new trade-off between symbiosis and immunity in the context of disease, and perhaps a new selective pressure on symbiotic relationships under this increasing disease and climate change. Now, I acknowledge that this still needs a lot of work, which is why we're moving ahead to do more analysis of the symbiont density experiment, our symbiont transcriptomes from all of those experiments, and then we would like to synthesize all of those transcriptomes into one model that will hopefully create a compelling link between transforming growth factor beta, symbiosis, and host immunity. And then eventually, I'd like to back this up with some model system experiments way down the line. So with that, I have a ton of people to thank. Tons and tons of people have helped me in the field. The Brandt Lab at the University of Virgin Islands, Ernesto Wales Lab at the University of Puerto Rico, Andrew Baker's lab at the University of Miami, and in particular, Ana Palacio, who was a huge help with that second experiment I talked about briefly. And then Vanessa Brinkhouse, who helped us with the Inicia disease and actually brought it to our attention. Here at UTA, the Casta lab, in particular Audra, and the Clark lab, Bob, were great helps with that pathway-based analysis I talked about briefly. And the Genomics Core facility has been an amazing help throughout my entire dissertation. Obviously, I have to thank my entire dissertation committee, Andrew Baker, who's here hopefully virtually, and then Todd, Jeff, and Matt, who have been a great help. I'm not going to cry. My amazing advisor, Laura, who has just been a great help this whole time and gone above and beyond professional development, and just, I wouldn't be here if it wasn't for her, and it would have been such an amazing dissertation. So thank you to her. And of course, I have to acknowledge my funding sources. I've been funded on the GRFP for the past 3 years, and also various grants from BioOsh and iOS have supported some of these projects, and UTA Phi Sigma has been an immense help. And then the fun acknowledgements. This one's dedicated to the Midlars Lab, because y'all are awesome. Obviously, my amazing advisor, Laura. The best undergrad one could ever ask for, Caleb, who's been such a trooper the past 2 years, helping me with so many random things. Contessa, who's been with me all but the first year, has been the best lab mate and office mate a girl could ask for. And then all of our more recent, Cindy, Brad, Nick, y'all have been great helps in past undergrads. Thank you guys so much. And then finally, I just have to thank all of my friends here and elsewhere, my emotional support animals and people, my family. Y'all have been great. I love you all. Thank you all for your help and support. And I will take questions. Yeah, Cindy?
I was curious, on your experiments where you're enriching the symbionts, are you focusing on particular genus or species of symbionts, or is it what's just already there?
It's what's just there, but it's predominantly type D. These are rescue corals, so they're kind of weird, but we do have data on what symbiont type they are, but they're all the same type. We didn't manipulate it at all.
I'm interested in the black disease.
Yeah.
It seems like from what you said that it's more as like a symptom of the cure than a symptom of the disease.
Yeah. We're still working to figure out what the etiological agent is.
Right.
But for most coral diseases, they're named or described based on the immune response, because we can't necessarily see what's going on inside them. We're confident it's some kind of a pathogen. We just don't know what pathogen.
Do you see those diseases resolve over time?
I have to ask our colleagues. I don't think we've seen any kind of.
Ernesto has, yeah.
Yes.
Our colleagues that are in the field a lot more than us.
Yeah.
You see some of them kind of scab off, at least with C-fans and stuff, they scab off and then regrow.
Okay.
If it doesn't take over the whole colony.
Yeah, a lot of times it's the entire colony that's covered in this response, so it's kind of like a futile attempt to fight off the disease, but
Right.
typically just ends in death.
It would be really cool to do kind of a similar analysis on something that has recovered.
Oh yeah. So Ernesto has been trying to do that with
Right.
different corals that have been gotten sick and recovered and multi-year. So hopefully eventually we'll have data on that. Yeah, Will?
So I'm very ignorant as to anything with feathers and have gills and fins, but you were doing your enrichments with ammonia, the non-ionized
Yeah.
corn. Is there toxicity associated with the ionized corn?
So I don't have the data today, but I have looked specifically at the effects of that nutrient treatment on gene expression, and they're very limited. So that's the nice thing about DEC, because I was able to model the effects of nutrient, the effects of immune challenge, and the effects of that continuous variable symbiont density all separately. And so I do have some data of none of these genes are affected by both. So I can go in and say what's affected by both and see what's a toxic effect, what's an immune challenge effect, and what's an effect of the symbiont density. Does that answer your question?
Yeah, but are the actual symbionts actually affected by?
They're increasing, so they're not negatively affected. They are increasing in density, so they're nitrogen limited. So this is a good thing, and they're breaking it down and increasing in abundance.
The only reason I brought it up is because ammonia will transfer between the forms depending on the pH and temperature conditions. So
these are kept very controlled. There's somebody in there every day checking on them and making sure that they're actually benefiting the coral and not harming it.
sweet.
Anyone else?
Any other questions? Okay, great. Let's thank Lauren again.
Thanks. Grab some more food if you want.
The defense will be in 118
128.
or 128. We're in right now. Hey.
Sorry.
I don't know what happened. I don't know what happened either. Okay, we're gonna we'll move into the defense room and then I'll call you back.
No, it's just
Okay.
Oh,
The Skype. Oh,
okay.
you set up.
I can set up
Okay,
a
she'll call you on Skype. I don't have Skype anymore or Facebook.
email. I have to email or text us to Skype things. I don't know if I have a Skype here.
Well, yeah, I think I don't have Skype on my computer.
I mean, well, that's also why.
Okay, I'll call you back.
Oh, what?
We go over with the computer. I was like, I need to focus. I don't know what's going on over there. No, it's fine. It's okay. No, I was just like, should I stop? Should I keep going?
The slides also look pretty good. Like it was working at first.
So, yeah, what happened? It just stopped.
Like the video was working at first.
Yeah. Because I looked and I saw it working.
Then afterwards, they're like, hey, so it says